Hello boys and girls, welcome back for another lesson of biology. Today we are going through lesson 35 of chapter 28, classwork 28.1 and the problem of GM organisms. Before you start watching the video, remember to like and subscribe my channel and also share the video to your classmates. Let's start. Please take out your classwork 28.1, let's go through it together. The diagram below shows how human insulin is produced using recombinant DNA technology. From the last lesson, you should be very familiar with this technology. It starts with the DNA encoding human insulin, that is the gene of interest from the donor cell, such as the insulin secreting cell in the pancreas. And you also need a plasmid, which is a vector to carry the gene of interest. The plasmid comes from bacteria. And after combining the plasmid and the gene of interest, you form the recombinant plasmid. And you put the recombinant plasmid back into the host cells. So now, these bacteria contains a new gene and also a new characteristics, so it can produce human insulin. First of all, question A. Can you explain the term recombinant DNA technology? It is actually a technology which a fragment of DNA or the gene of interest from a donor cell is isolated, get it out, and then inserted into the DNA of another organisms or another cell. So let's write it down together. For question B, what is a recombinant plasmid? As you can see, a recombinant plasmid consists of a plasmid and also the gene of interest going together. So a recombinant plasmid is a plasmid formed. It is a plasmid It is a plasmid formed after the gene of interest from the donor cell is inserted into the plasmid. So this is the meaning of a recombinant plasmid. Question C, state two enzymes that are required in the production of the recombinant plasmid and give the function of each enzyme. So you have learned two different enzymes. They do different works in the preparation of the recombinant plasmid. The first one is an enzyme for cutting. The second one is an enzyme for combining and sticking. So the first one, we call it the restriction enzyme. What is the function of restriction enzyme? It is used to cut the gene of interest. This time it is actually the human insulin gene. And also to cut open the plasmid to give on both ends a sticky end. And for the second enzyme that is used in the preparation of recombinant plasmid, it would be an enzyme for sticking and joining. We call it a DNA ligase. So the function of it is to catalyze the joining of gene of interest and the plasmid. And then question D, describe how the human insulin can be produced using the GM bacteria. After the human insulin gene goes into the bacteria will try to express the DNA or the gene and using the bacteria's own machinery to produce these protein. So let's write down. The human insulin gene is expressed in bacteria. And insulin is produced using the machinery. The machinery of bacteria, that means the machinery or the steps and process of protein synthesis in the bacteria. Last question, question E. What is the medical use of human insulin? It is for blood glucose level control in the diabetic patient. So they are used in injection. It is injected to diabetic patient to control blood glucose level. For the second part of this lesson, we are going to talk about the problem caused by GM organisms. What is the consequence of cultivating GM corn plants with insect toxin? Nowadays, the farmers, they will grow some type of plants which is genetically modified. There is an insect toxin gene inserted into the fruit or the plant. So now the plant itself, it can release the insect toxin in order to kill the pest. 害蟲 such as 蝗蟲啦, so they will eat up a lot of plants, so leads to a very low production of plants and vegetable, etc. So it helps to kill the very damaging, annoying pests. However, if you GM the plants and allows them to release some insect toxin, sometimes not only will it kill the pest, it will also kill other species, other insect species, and leads to extinction, 绝种. And now, not only leading to extinction, these insect toxins, they might also kill some beneficial insects, such as bees, 蜜蜂. These bees, 蜜蜂啊, they are very helpful in pollination of plants. If you kill such beneficial insects such as the bees, which helps in pollination, there will be less insects helping in pollination and reproduction of flowers. This can be very troublesome. Not only will the insects be extincted, some plants or some flowers which relies on the pollination by the bees, they will also get extincted. So this is a not good situation. This is a problem. Another problem caused by the genetically modified organisms, the GMO, might be ecological disaster or ecological imbalance, 生態災難或者生態不平衡. So this is because a few years ago, there's a news given by Professor Zhang. 張馬山博士咁啊，張馬山博士咧，佢係我哋香港呢個生態教育及資源中心科學事務嘅經理嚟嘅。咁咧，佢特別喺呢個香港嘅保育方面咧，有好大嘅貢獻啦嘛，間唔中都有啲講座啊或者活動咧，提供俾我哋香港市民同埋一眾同環保有關人士嘅。咁啊，佢講咗一個咩新聞咧？就係原來咦30蚊一條嘅沙巴龍躉點解可以咁平咧？原來沙巴龍躉咧就唔係一個正常嘅物種嚟嘅，佢係人工混種出嚟。咁啊，都可以話一種 GMO 啦。咁啊，食量非常驚人，亦都可以咧好極速咁生肉長肉，變成一條好大條嘅魚。但係咧有機會咧引發出呢個生態災難。咁啊，點解咁講咧？原因我哋睇下沙巴龍躉係咩嚟嘅先。沙巴龍躉咧其實係花尾龍躉同埋老虎斑經人工嘅方法混種出嚟嘅魚嚟嘅，本來係唔應該存在喺大自然。咁其實佢哋都係一種 GMO 啦。咁佢哋嘅死亡率咧極之低。而且食量係非常驚人，又會獵食大量嘅細魚啦，咁啊爭奪本地魚類嘅資源咧，嚴重威脅佢哋嘅生存啊！咁啊，我哋學過啦，佢一種新嘅 species 咧，可以令到個 competition 大增啦，咁都爭晒人哋啲 resources 喎，咁一啲細魚咪冇埞企咯，好易死啦！咁啊，呢條大約長20幾厘米嘅沙巴龍躉咧，生長嘅時間就係需要1年。但同樣大小嘅青斑咧，要生到20幾 cm 咧，通常係要2至3年嘅。咁所以呢一種沙巴龍躉咧，佢係一種GMO 咗嘅生物咧，佢生長特別快啦，咁亦都咧 competition 上面咧，佢亦都贏啊。咁所以咧有機會咧導致咧本地應該有嘅一啲細魚仔啊 local 嘅一啲 species 咧，就會出現一個唔夠爭嘅情況，會輸啊。咁啊有機會出現到生態不平衡或者生態災難。Alright, so that's it for the part about the Classwork 28.1 and also the problem caused by the genetically modified organisms, GMO. I will see you in the next lesson. Bye-bye.